Imagine for a moment having the power to actually alter the blueprint of human existence. I mean, what if we could just wipe out inherited diseases? Or maybe enhance our own capabilities, even influence things like intelligence.
It really does sound like science fiction, doesn't it? But it's not anymore.
Exactly. This is the frontier of genetic editing, a power that really forces us to ask, are we actually ready to rewrite the code of life?
It's a huge question.
It is. And today, we're diving into a really remarkable book about this life sciences revolution. It focuses on the incredible journey of Jennifer Doudna and the gene editing tool CRISPR.
Right. And the goal here, I think, is to pull out the crucial insights, not just the science, which is amazing, but also the deep ethical and societal questions that come with it.
Absolutely. And as we get into this, remember the aim is always to spark reflection, maybe some rational thought. For more analyses like this, powered by AI, looking at other groundbreaking books, you should check out Mindful Moments on our YouTube channel.
It's definitely a pivotal moment we're living through. And understanding how we got here is, well, it's key. The book does a beautiful job showing how Doudna's early curiosity was really that initial spark.
Yeah, tell us about that. Her childhood sounds fascinating.
It really was. Her fascination with nature, those walks in Hawaii she took with a biology professor, that apparently ignited her passion for understanding, how and why things worked at the most fundamental and inner level.
So it started really young. How did that kind of curiosity, seeing science as this mystery she wanted to solve, prepare her for something like CRISPR?
Well, I think the key insight is how these different threads came together. Her interest in nature's workings, but also this humanistic view she got partly from her father. I mean, she wasn't just focused on the code, but also on what it meant for us, for humans.
Right.
Reading the double helix when she was 12 was apparently a huge moment. It made science feel like this thrilling puzzle, not just emotionless men in lab coats, as she put it. Mm-hmm. But it wasn't always easy for her. She struggled a bit at Pomona College initially, felt out of place. But the book says that experience actually stiffened her resolve. And that love for the humanities her father instilled, it became really crucial later for tackling those big ethical questions around CRISPR.
So she had this blend of scientific drive and humanistic thinking. And this was all happening as the world itself was shifting, right? Moving from the information tech era into this new life science revolution.
Exactly. Building on decades of work.
Yeah, you had Mendel back in the day with his pea plants showing traits don't just blend together, they're dominant or recessive.
Foundational stuff.
Totally. And then Watson and Crick uncovering the double helix structure of DNA, that beautiful structure that showed how life could copy itself.
And let's not forget Rosalind Franklin's X-ray images, which were absolutely vital for that discovery. Even brilliant people like Linus Pauling had, bloopers, like his incorrect triple helix model.
Right, science isn't always a straight line. And then came the Human Genome Project, this massive international effort. There's an interesting, maybe slightly uncomfortable connection back to Data's childhood hero, James Watson, isn't there?
There is, yeah. The book touches on how Watson's son Rufus and his personal struggles later brought up some difficult history related to the eugenics movement, particularly concerning Cold Spring Harbor, where Watson was director. It's a reminder that science doesn't happen in a vacuum. It has history, sometimes a complex one.
A really complex history. Okay, so back to Doudna's path. RNA. It was seen as just this, dull intermediary for a long time.
Yeah, the messenger molecule.
But it became her focus in her doctoral research. Why was her mentor, Jack Szostak, so convinced RNA held these huge secrets about the origins of life because of its catalytic powers?
Exactly. Szostak saw something others missed. He realized RNA wasn't just a messenger. It could act like an enzyme, a catalyst, that maybe was central to how life even began. And Doudna, she really embraced collaboration. The book highlights how she'd mentor younger researchers, even putting undergrad as lead author on a major paper. That team spirit was pretty central later on.
That spirit seems crucial. Now let's talk about CRISPR itself. You called its unveiling revolutionary. Take us back to the beginning, these almost accidental discoveries.
Well, it started with something that seemed pretty minor. Francisco Mojica, a scientist in Spain, noticed these weird, mysterious repeated sequences in bacteria.
Yeah.
He actually had trouble getting funding for it. People told him it was probably nothing special.
It happens all the time in science, right?
It does. But then came this eureka moment at Danisco, a dairy company of all places. Philip Horvath and Rodolphe Barangu were looking at bacteria used in yogurt production. Using computational tools, they compared these CRISPR sequences in the bacteria to viruses that were attacking them. And bingo! They realized the bacteria were basically grabbing snippets of viral DNA and storing them in their own genome
Yeah.
in these CRISPR regions.
Wow, like a genetic vaccination record?
Kind of. It was bacterial immunity passed down through generations. They found a perfect match between the stored sequence and the attacking virus. It was incredible, like the bacteria were remembering their enemies.
So they had this microbial most wanted list. What happened when that finding got out? Was it an immediate aha moment for everyone?
For people in that specific field, it was definitely a holy cow moment, as the book puts it. But the really critical next step came from Luciano Marafini and Eric Sontheimer. They figured out that CRISPR targets DNA, not RNA.
Ah, okay.
Right.
Why was that so significant?
Because if it cuts DNA, it meant, well, the implication was huge. It could potentially be harnessed as a tool to edit genes precisely. That finding really paved the way for the Doudna-Charpentier collaboration.
And that collaboration is legendary now. Tell us how Jennifer Doudna and Emmanuel Charpentier actually connected.
It was pretty serendipitous, actually. They met at a conference in Puerto Rico in 2011. Very different personalities. Apparently, Doudna with her intensity and sly humor. Charpentier more quietly elegant, but they clicked immediately.
And they started working together.
Intensely. The book calls it a model United Nations effort. They were collaborating across time zones, mostly via Skype, this constant 24-hour push to figure out the mechanism of CRISPR-Cas9. Cas9 being the enzyme, the molecular scissors, that does the actual cutting.
Incredible dedication. What was the key breakthrough that turned it from an interesting bacterial system into a usable tool?
That came from Martin Janek, a postdoc in Doudna's lab, working closely with her. They had this brilliant idea. What if they could fuse the two separate RNA molecules that guide the Cas9 enzyme, the crRNA and the trachRNA, into one single guide?
Simplify it.
Exactly. They created this elegant single guide RNA. Think of it like this. Cas9 is the scissors, and this single guide RNA became the programmable GPS. You can now easily tell the scissors exactly where in the vast genome to make a cut. It made the whole system much easier to program and cheaper. Infinitely programmable, really.
And they knew right away how big this was. I read about a celebratory dinner.
Oh, absolutely. They had dinner at Chez Panisse, a famous restaurant near Berkeley. They knew they were at the start of something immense, this intense time when science was all coming to fruition. And they started thinking, even then, about the implications.
Which, of course, led eventually to the Nobel Prize.
Indeed. The 2020 Nobel Prize in Chemistry, shared by Doudna and Charpentier, for developing a method for genome editing, rewriting the code of life.
A truly historic moment. But, as often happens with big discoveries, the competition heated up fast, didn't it? The rivalry between Doudna's group and Feng Zhang's group at the Broad Institute.
Yeah, it got pretty fierce. Patent disputes, arguments over who did what first, especially when it came to applying CRISPR in human cells, which Zhang was really pushing for. That patent fight is actually still ongoing, trying to figure out if Zhang's work on human cells was a distinct invention from the original discovery.
And there was that Heroes of CRISPR controversy, too.
Right. Eric Lander, a very prominent scientist, wrote a piece that many felt minimized Doudna and Charpentier's roles. It caused quite an uproar. Lander later suggested his own motivations might have been complex, referencing a play about scientific ambiguity. It shows how much ego and narrative can play a role.
But it wasn't all conflict, was it?
No, and that's important. Especially later, during the pandemic, there were moments of real cooperation between these competing labs. So competition was still there, but maybe less cutthroat when facing a global crisis.
Okay, so from the lab benches and the rivalries, let's turn to the really deep ethical questions. Curing diseases like Huntington's or sickle cell, the book mentions David Sanchez, a teenager suffering terribly from sickle cell. That seems like a clear win, right?
Most people would agree, yes. The potential for treating devastating genetic diseases is immense.
But then you get into the gray areas. What about enhancing traits, making people stronger, like those mighty mice experiments, or maybe even trying to tweak IQ?
Or height. The standing on tiptoes problem, where if everyone gets taller, nobody really gains an advantage. Or even changing eye or skin color. It quickly moves from medicine to something else entirely.
It does, and it brings up this continuum conundrum. Is editing genes that get passed down, germline editing, fundamentally different from, say, prenatal screening, which we already do?
That's a huge debate. Some ethicists argued it could even be a moral imperative. The right of every child to be born with a sound physical and mental constitution.
While others warned about playing God.
Exactly. People like Paul Ramsey, Jeremy Rifkin, they raise alarms about designer babies, a Brave New World scenario. Michael Sandel, the philosopher, talks about the danger of losing our appreciation for life as a gift if we strive too much for mastery and control over our own biology, undermining empathy, perhaps.
And what about disability? The book mentions a deaf couple who specifically wanted a deaf child, seeing deafness as part of their culture, not something to be fixed.
Right. It challenges the very definition of normal or desirable. And then there's the massive issue of inequality. If these enhancements aren't free, and they certainly won't be cheap, could they greatly increase inequality and indeed encode it permanently in the human race?
That's a chilling thought, creating a permanent genetic class divide.
It's one of the biggest concerns.
And this ethical minefield just exploded with the CRISPR babies incident, didn't it?
Oh, absolutely. In 2018, He Jiankui, a Chinese scientist, just announced he'd created the world's first gene-edited babies twins, edited supposedly to be resistant to HIV.
Which wasn't even medically necessary, right? There are other ways to prevent HIV transmission.
Exactly. It seemed reckless, and it violated all the international guidelines that were being discussed. The scientific community was outraged. Doudna, Matthew Porteous, many others condemned it.
Why did they think he did it?
The general feeling, reflected in the book, was that it stemmed from this intense pressure in science to be first, to be famous, this culture-rewarding, provocative research celebrity, national scientific competitiveness.
So the backlash was huge.
Global backlash. It led to calls not necessarily for a complete ban or moratorium on inheritable editing, but for a much more cautious, responsible pathway. Doudna herself argued against a moratorium, saying the genie was kind of out of the bottle already. Her view largely shaped the international recommendations that followed, focusing on a prudent path forward, only for serious diseases under strict oversight.
And then almost right after that shock, the world faced another crisis.
Yeah.
The pandemic.
Yes, and suddenly CRISPR wasn't just about long-term ethics. It became an urgent tool for right now.
How did that shift happen?
It was incredibly fast. Labs like Doudna's Innovative Genomics Institute, Zhang's Broad Institute, they pivoted almost overnight. The immediate problem was testing. There was this fire-on-my-ass problem, as one scientist put it. The U.S. government's initial testing rollout was slow, flawed.
I remember that. It was chaotic.
It was. And universities basically had to step into the breach, setting up massive testing operations. It was amazing mobilization.
And CRISPR played a role in the testing itself.
Yes, absolutely. Researchers quickly developed new diagnostic tests based on CRISPR technology, like Detector and Sherlock. These could detect the virus's RNA very specifically and potentially much faster and cheaper than existing methods. It showed CRISPR's versatility.
And vaccines, too. The RNA vaccines were a game changer.
Huge game changer. The pandemic massively accelerated the development of genetic vaccines, especially RNA vaccines from companies like Moderna and BioNTech Pfizer.
Why were RNA vaccines so suited for this?
Well, a key advantage is speed. RNA vaccines deliver instructions directly to the cell's protein-making machinery in the cytoplasm. They don't need to get into the cell's nucleus where the DNA is stored. That simplifies things and allows for much quicker design and production compared to traditional vaccines or even DNA vaccines. It truly transformed vaccine development.
It felt like science fiction becoming reality almost overnight. And this push wasn't just in huge labs, right? The book mentions biohacking.
Yeah, Josiah Zahner comes up again. He's known for some provocative stuff like DIY gene editing kits. During the pandemic, he was involved in efforts to sort of democratize the science, sharing vaccine information, showing that even smaller groups, a bunch of scrubs like us, as he put it, could contribute.
So bringing biology into the home, almost like personal computers did for digital tech.
That's the analogy the book draws, this idea of citizen science empowering more people to understand and even participate in biotech. The pandemic definitely shifted perspectives. Suddenly, the idea of editing our own genes, maybe for immunity, seemed, well, less appalling and a bit more appealing to some.
Which brings us back to that core dilemma. After billions of years of evolution working, naturally, we humans can now effectively hack the code of life. We can engineer our own future.
Is it unnatural? Is it playing God? Or is it just another step in evolution?
Yeah.
Maybe nature, as the book speculates, evolved a species that could actually take control of its own genome.
That's a wild thought.
It is. And the fundamental question left for you, the listener, isn't really can we do this anymore? It's how will we? How do we as a society wisely navigate this incredible, almost terrifying power to shape what our species becomes.
Will it lead us to thrive, or could it, like some evolutionary dead ends, put our survival at risk?
Evolution doesn't always pick winners in the long run. It's a profound responsibility.
It truly is. So this exploration of CRISPR shows us this incredibly powerful tool, born from pure curiosity, honed by competition and collaboration, and now presenting us with these enormous ethical choices about our own future.
A lot to think about.
Definitely. For more analyses like this, digging into complex topics from insightful books, remember to check out Mindful Moments on our YouTube channel and explore our other podcasts as well.
Keep questioning.
Absolutely. Subscribe to this discussion so you don't miss future explorations. Share it with someone you know who loves to think critically. And if you want to go deeper into the story of CRISPR and its implications, consider getting the featured book. You can find a link in the description, along with links to other books we discuss. Your engagement really helps us keep bringing you these kinds of insights.
Until next time.
Keep questioning, keep learning, and keep reflecting.